Hi, I'm Catherine Mikulinski, and I'm going to be walking you through the mitosis meiosis lab this week. I wanted to do this short introductory video just to let you know what to expect and what materials you want to have on hand before you begin. Of course, you're going to need your lab. So this lab begins on page 105 in your lab book, and I really want to stress with you how important the questions at the end of this lab are. They really are going to synthesize the work that you've done during the lab, and they'll really let you know if you know what you're doing or not. The second thing I recommend that you have is this two-page handout. Now have this open on another screen while you're working on your questions. It's just a really nice image of mitosis and meiosis stages. Really helpful to have a really nice figure like this, very clear, especially when you're answering the questions. The third thing you're going to need is something that you can use to represent chromosomes. So in lab, we would be using poppet beads. They look like this. I'm holding the centromere, and then here's the chromosome. There you have it. These are great. Unfortunately, you probably don't have poppet beads at home, so what can you do? Well, you just need to make your own chromosome. So you might want to think about pipe cleaners. You could think about yarn. Perhaps Play-Doh would work really well, I think. And you might even want to cut them out of paper, just so that you have the chromosomes you need to manipulate to help you really get a handle on the different stages of mitosis and meiosis. So just want to refresh your memory a little bit. Remember that chromosomes come in pairs in diploid cells. Our diploid number is 46. Each one of your body cells has all 46 chromosomes. And why do we use the word diploid? Di means two and ploid means set. And so diploid cells have two sets of chromosomes in some sense. In other words, they have two chromosomes for each pair. So here we have two long ones, one you got from your mom, one you got from your dad, and then we also have a pair of short ones, one you got from your mom and one you got from your dad. If the chromosomes were to ravel up or to condense during gap one, this is what they would look like. So this is what the chromosomes in the cell typically would look like. So that's what the starting cell looks like. It is a diploid cell, two chromosomes of each type. So the diploid number for our demonstration cell is 4. 2 pairs, 2 times 2 equals 4. Now, chromosomes normally look like this, but recall that before a cell divides, you need to make a copy of the DNA, and we call that copy a sister chromatid. So here is a duplicated chromosome, a chromosome that has 2 sister chromatids. And the only time you're going to see that second chromatid is when the cell is getting ready to divide. It's gone through the S phase of interphase and duplicated the DNA to form the sister chromatid. So here's our same demo cell, still with 4 chromosomes, but now 4 duplicated chromosomes, a pair of short ones and a pair of long ones. Physically moving these through the stages is really going to help. I also think it might be important if you try to work together, remotely of course, because it can be really, frankly, challenging material. And sometimes, remember, it sounds easy when someone like me is talking about it, but it's still very difficult material. So I hope you do okay. I'm working your way through the rest of the videos. I also have posted just the straight PowerPoint without the narration. So good luck. Probably see you again. Bye-bye.